Titin is pleased to present this video by Dr. Wolfgang Linke, Professor and Chair of Physiology at the University Hospital of Münster in Münster, Germany. Dr. Linke was unable to join us live for the 2022 SciFam conference, but he gave us permission to use this video, which is publicly available on the Touch Cardio website. He shares his thoughts on the Titin gene, truncating variants, and dilated cardiomyopathy from the virtual ESC Congress 2021.
I'm Wolfgang Linke, Professor and Chair of Physiology at the University Hospital of Münster, Germany. The Titin gene is one of the largest genes in our body. In fact, the Titin protein is the largest protein in our body, and it happens to be in muscle cells. And in these cells, the Titin is important for providing a template, like a blueprint for the contractile units of the heart. In other words, without Titin, there would be no heart contraction. And we do know that mutations in the Titin gene called Titin are well known to cause a severe heart disease called dilated cardiomyopathy. And in fact, we have learned that depending on the population patient cohort studied, between 15% and 30% of DCM patients get the disease due to a Titin mutation, a specific type of Titin truncation called truncation, where one of the two Titin alleles is shortened, is truncated, and this is causing this severe disease. The only cure for dilated cardiomyopathy is there is no cure. The only solution is a heart transplantation. Titin is best known for being an elastic protein. So it provides the elasticity to the cardiac walls, together with other structures such as the extracellular matrix consisting of collagen and other proteins. But Titin is a protein of the contractile units called the sarcomeres. And without Titin, there would be no elasticity or a little elasticity of these contractile units. So any modification to Titin, such as by a mutation, would affect the cardiac filling, so the filling of the chambers of the heart with blood. And because the filling of the chambers of the heart is correlated to the cardiac output, to the pump function of the heart, via a well-known mechanism called the Frank-Starling law, also the active contraction, the contractility of the heart is affected if the filling is not proper. So these are two of the main properties, main functions of Titin. And in addition, as I mentioned before, Titin is a template for these contractile units. And without a proper Titin protein, there will be no contractile units in the cardiomyocytes and no contraction. TTN-TVs, name implies truncating variants in the Titin gene, are the best studied mutations in the Titin gene in connection with cardiomyopathy. In fact, TTN-TV do not only cause dilated cardiomyopathy, they also cause other types of cardiomyopathy, including acquired cardiomyopathies, such as alcohol abuse-induced cardiomyopathy or cancer treatment-induced cardiomyopathy. And we know that in all these cases of cardiomyopathy, there is a certain region in Titin gene which is most frequently affected by these mutations. And this is, in fact, exactly the template region for the contractile units. We call this the A-band region. And the reason for this is that this region is expressed in all Titin proteins that we know, in all big Titin proteins that we know. We call this constitutive expression. It's always there. And because it's always there, any mutation that affects this region will be quite severe, affecting the function of Titin. So let me summarize. All regions in Titin that are constitutively expressed, that are always expressed, the mRNA and exon level, protein level. If the mutation, if the TT-NTV occurs in these regions, it will have a severe effect on the health of the patient. This is a heavily studied question in the field, and recent work has provided important insight into these novel, let's say, novel pathomechanisms of TT-NTV. So what happens in these patients is that because there is only one healthy Titin allele left and the other is truncated, the question is whether this one allele is enough to make enough healthy Titin protein. And our work has shown, or recent work has shown, that this is not the case. These TT-NTV DCM patients have less normal Titin protein than control groups. We call this haploinsufficiency, Titin haploinsufficiency. And if there is less normal Titin, it is also clear that there are less contractile units in these hearts. And this causes the reduced contractility of TT-NTV. This is the first pathomechanism, Titin haploinsufficiency, causing loss of contractile units. The second pathomechanism, which is quite striking because it has been overlooked before, but we could show that recently, that if there is a truncated mRNA, a truncated transcript, one Titin allele, the question is a truncated protein made? Because quite often a mutated protein is attacked by the protein degradation machinery, the cardiomyocytes, and is not visible anymore. But in case of Titin, we found that truncated Titin proteins are stably expressed in the cardiomyocytes. And they could act as poison peptides because we found that the younger the patient at the time at heart transplantation, the more truncated Titin protein contained in the sites. So we think these truncated Titin proteins are disease relevant. These truncated proteins do not build into the contractile units because they are truncated and they cannot provide a template for the sarcomere. But they are aggregating in the cardiomyocytes. So we found aggregates containing these shortened Titin proteins in the cardiomyocytes of the affected patients. We have, again, in a recent study, addressed this issue by using disease modeling in cultured cells, a specific type of cultured cells. In fact, we took tissue, in fact, skin tissue, from a healthy patient and reprogrammed this tissue in cell culture to make stem cells, so-called pluripotent stem cells out of this tissue. And from a pluripotent stem cell, we can make all sorts of different cell types again. And in our case, we made human cardiomyocytes. We cultured human cardiomyocytes out of these stem cells. And then we introduced a Titin truncation in these cell cultures using the gene scissors CRISPR-Cas9. So we performed gene editing and introduced the truncation in cell culture. And we could very nicely confirm the described pathomechanisms. And we could also test what type of treatment would be useful to correct the contractile function of these cells. Because we also performed the same type of cell culture with patient-derived cells, so where we took a tissue from a TT-NTV heart patient, and then we repaired, using again the gene scissors CRISPR-Cas9, we repaired the truncation. And we found that in the case of truncation, contractility of these cells is impaired, but it can be fully rescued by correcting the truncation. So the best solution for the patient would be to repair the truncation using gene editing, which is still very difficult, if not impossible, in patients at the moment. But it works well in cell culture. And of course, there's a lot of research in the field. And we hope that at some point we will be able to treat patients using gene editing, or more extensively using gene editing. And another way we tested, another option for potential treatment, is to use modulators of the intracellular protein quality control systems, because protein quality control in these cells is impaired. As I mentioned, a lot of truncated titin protein is present, and that impairs the degradation machinery, the protein degradation machinery in cells. And when we modulated this protein degradation machinery, we could also improve the function of these, contractile function of these cells. I think for some people it's quite interesting because these TTNTV mutations are quite common even in the healthy population, and but not all of them get sick. Obviously, most of them are healthy. That's why it's called a healthy population, or apparently healthy. So we have understood from previous research and our recent research that TTNTV represent a main risk factor to get a heart disease later in life. If the TTNTV comes together with an additional stressor, such as wired stressor, for example, high cholesterol, or high blood pressure, or diabetes, or obesity. So these are all risk factors. And if they fall on the background of a TTNTV genetic modification in the patient, then the chances are much higher for the patient to get the heart disease.